COVID-19 vaccine efficacy is on the top of everyone's mind. Pfizer and BioNTech are moving along with FDA approval and showing upwards of 95% vaccine efficacy. Moderna is underway as well with 94.5% efficacy, and now Oxford and AstraZeneca seem to also be gaining some traction. In a previous video, I explained what those incredibly high efficacy numbers mean, and in this video, I'm going to help build on that intuition by walking through the intuitive answer to the number one question I received in response to that last video. How can we conclude much when the number of infected individuals in the vaccine trials was so low? For the Pfizer trial, at least for the preliminary data, only 94 people out of nearly 39,000 were infected with COVID, and yet everyone around the world was celebrating the trial's results. For the Moderna trial, only 95 people out of about 30,000 were infected, and we had the same basic celebratory conclusion. So how is it that, with only 90 or so people infected out of 30,000, we can be so sure that the vaccine actually works? Welcome to Data Demystified. I'm Jeff Galak, and in this episode, I'm going to go through the intuition for why even seemingly small sample sizes like this can often tell us a whole lot. If you stick around, I'll quickly go over the results of the vaccine trials and then work to build your intuition for why even with 90 or so infections out of tens of thousands of research participants, we should feel confident that the results of these trials are actually very strong evidence in support of the efficacy of those various vaccines. To keep things simple, I'm just going to focus on the Moderna results since the basic story is the same for Pfizer. Anyway, this vaccine trial enrolled 30,000 people, and when they hit 95 infections, they released their preliminary results. To be sure, they're still collecting more data before submitting for the FDA approval, but those initial results are what everyone is so excited about. What's critical is that Moderna conducted a double-blind, placebo-controlled trial. That means that half of those 30,000 people were given the vaccine, and the other half were given some harmless saline solution, also known as a placebo. Of the 15,000 who got the vaccine, only 5 were infected with COVID-19. And of the 15,000 who got the placebo, 90 were infected with COVID-19. This is where we can compute the reported efficacy of 94.5%, something I cover in my other video. Okay, a quick aside. I had an error in my previous video where I explained how to compute efficacy. To be clear, the intuition is still there. Efficacy tells you the relative rate of infection across the vaccine and placebo conditions, and not how likely you are to get COVID if you were first vaccinated and then exposed to COVID. But I did make a mistake, and I absolutely want to own up to it. I incorrectly said that efficacy is computed as the number of infections in the placebo condition divided by the number of total infections. That's not correct. Assuming equal numbers of individuals across the vaccine and placebo conditions, efficacy is computed as 1 minus the number of infections in the treatment condition divided by the number of infections in the placebo condition. Again, the intuition doesn't change, but the calculation does, if only ever so slightly. Anyway, because there were far fewer infections in the vaccine condition compared to the placebo condition, with an efficacy of 94.5%, Moderna concluded that their vaccine was really quite good. But before we celebrate, we need to ask ourselves, could those same results be due to chance rather than due to the vaccine? In other words, if we got 30,000 people together, flipped a coin for each to assign them to one of two groups, and then just let those groups go about their lives with no one getting a vaccine, could we get the same result that Moderna got? One group with 5 infections and the other group with 90 infections. Another way to think about this is, could we get the same result if the vaccine really did nothing at all? Because if we could get that result just by chance, that would seriously undermine our confidence that the vaccine is what caused that big difference in infection rates. This is where statistical significance testing comes in, and that's something I cover in a different video that I'll link to below. But significance testing in this case is actually not at all intuitive. Indeed, unless you are immersed in statistical significance testing for your education or your career, there's little that I could do to explain the intuition behind all of this using things like z-scores and p-values. Even I have a hard time with that in this case. Instead, I'm going to take what I think is a much more intuitive approach, simulations. So here's what I'm going to do. I'm going to build a simulation that tries to see how likely it is that we observe that result, 5 infections with a vaccine and 90 infections without, if the vaccine were actually completely worthless. In other words, we're going to build a made-up world where we take 2 groups of people, randomly infect some of them with COVID-19, and see how likely it is that we get that split of 5 and 90 in our 2 different groups. Here's how this works. We'll make a few simple assumptions, and then we can change those assumptions afterwards to see what happens. To start things off, I'm going to mirror the Moderna study as closely as possible. So we'll have 30,000 people, half in our vaccine group and half in our placebo group. And across all 30,000 people, we'll have 95 of them get infected. What's important though is that the way the simulation is built is that the vaccine in our made-up world doesn't work at all. So someone in the vaccine group is just as likely to be infected with COVID as someone in the placebo group. So what we're doing is seeing how likely it is to get that same 94.5% efficacy value just by chance, since in the simulation, vaccines provide absolutely no protection from the virus. To do that, for each person, we're going to pretend we have a big jar filled with 10,000 balls in it. Since the infection rate of 95 people out of 30,000 is 0.3%, we're going to color 30 of those balls red and the rest of them green. And we'll pretend that each person picks a ball out of the jar. If they get a red one, we'll say that they're infected. And if they get a green one, we'll say that they aren't. Then we repeat that for all 30,000 people, half who are in our vaccine group and half who are in our placebo group. Though, remember, vaccine here doesn't really work and do anything at all. If we do that just once, we find that 45 of 15,000 people in the vaccine group are infected, and 50 out of 15,000 people in the placebo group are infected. That translates to an efficacy of just 10%, which is not so good. To be clear, the true efficacy of the vaccine is actually 0. But by dumb luck, a few extra infected people wound up in the placebo group, and so it looks like the vaccine worked a little bit. But in reality, we know it doesn't work at all. To fix that problem of bad luck, rather than running the simulation once, we can repeat it a whole lot of times. In fact, let's see what happens if we repeat the simulation 100,000 times. Before we do that though, if you like what you're seeing, I'd really appreciate it if you could like this video, subscribe to this channel, and click that little bell icon so that you don't miss out on any new content that I put out. With that said, let's see what our 100,000 simulations show us. So every time we run a simulation, we can compute the efficacy like we did in the first example. And then we can plot the distribution of efficacy like this. This is called a histogram, and just shows us how often each efficacy value appeared in each of our simulations. Let me quickly walk you through this so that you can get a better sense of what this all means. The height of each bar tells us how often the simulation showed us an efficacy in the range displayed on the horizontal axis. The first thing to notice is that of our 100,000 simulations, most of them show an efficacy pretty close to 0%, and that makes perfect sense, since, well, we created a simulation to reflect a vaccine that doesn't work, and that's exactly what we see most of the time. But sometimes, just dumb luck gets in the way, and our two groups differ in terms of infection rates. For example, here you see that about 5% of all of our simulations had efficacy rates between -30 and -40%. That's right, in those simulations, the vaccine group had more infections than the placebo group. And just by chance, we'd expect that in a world where the vaccine doesn't work at all. But what really jumps off the screen is this part here. This is what Moderna found, 94.5% efficacy. And what you see is that in our 100,000 simulations, where we assume that the vaccine is worthless, we never see a 94.5% efficacy. Not even close. In other words, if the vaccine were actually worthless, with 95 observed cases of COVID-19 across all 30,000 people, the likelihood that we'd see 90 of those cases in the control group is virtually non-existent. But this is where you might be thinking something along the lines of, well, just to be safe, what if we waited a bit longer for more people in the trial to get infected? Wouldn't that give us even more confidence in the result? And the answer is that if the 94.5% efficacy rate stays the same as the number of people in the trial increases, you would absolutely have more confidence. Let's say that we waited till 1,000 people were infected. We can rerun our simulation now with 1,000 people infected instead of just 95. Well, now we get this new histogram. When we have 1,000 infections, the distribution really contracts towards the middle. And that's because it would take a lot more bad luck to see anything other than a 0% efficacy. That means that if the vaccine is ineffective and we had 1,000 infections out of 30,000 people, in almost all cases we'd get an efficacy close to 0%. That also means that getting an efficacy of 94.5% is basically impossible in this case. On the other hand, it would take us a lot more time to get to this point, perhaps prolonging how long we'd go without an actual vaccine. We can also go the other way and say, well, what if we didn't want to wait that long at all? What if we had only 40 people infected out of 30,000? What would that look like? Well, now the distribution gets wider, because it takes even fewer people to move from the placebo group to the vaccine group to make it look like there was a relatively high efficacy. There's just more room for a little bit of bad luck to make a result deviate from the truth, which in this case, and in this simulation, is that the vaccine just doesn't work at all. This is all to say that even with these tiny infection numbers, we should have a lot of confidence in the efficacy of the vaccines. It's just super unlikely that we'd see 90 infected people in the placebo group and 5 infected people in the vaccine group if the vaccine really didn't do anything at all. So what would it take to get that type of result? Well, the beauty of simulations is that we can change the underlying assumptions. In the simulation so far, I've assumed that the efficacy is actually 0, and we saw what the distribution of results would look like, just due to some random fluctuation in the data. Well, what if we tell the simulation that the efficacy should be, say, 10%? That would mean that the vaccinated group would have 10% fewer infections on average than the placebo group. But how likely would we be now to find something like a 94.5% efficacy? Looking at this distribution, it would still be super unlikely. That is, if the true efficacy of our vaccine is only 10%, it's really unlikely we'd observe an efficacy of 94.5%. Well, what if we bump that up to 50% efficacy? Now we're getting close, but it's still really unlikely to see a 94.5% efficacy if the true efficacy of the vaccine is only 50%. So of course, what if we set the simulation efficacy to 94.5%? Well, here you see that if the vaccine really has a 94.5% efficacy, out of 100,000 simulations, we get an efficacy estimate somewhere between 79 and 100%. But in case you're wondering, that lower bound of 79% is actually really uncommon. In fact, of those 100,000 simulations, over 85,000 of them come back with an efficacy between 90 and 100%. In other words, when we see a result in the vaccine trial, like 94.5% efficacy, it's really likely that the true efficacy of the vaccine is somewhere around 94.5%. The big takeaway from all this is that even with a small number of infections, given how almost all of the infections, 90 out of 95, were in the placebo group, we should feel very comfortable with the fact that the reason that the difference across groups emerged is because of the vaccine, and not because of chance. Before ending, I do have a request for all of you. If you're someone who's good at putting together simple web apps and would be interested in helping me make the simulation I made interactive, I'd love your help. Right now, I use some pretty cobbled together PHP code to get this all to work, entirely because I happen to know PHP pretty well, even though PHP is definitely not optimized for this. So yeah, if you want to help me out, I can share all my code with you and work through how to make the simulation work. If we can do that together, we can share the interactive simulation with the world and help build everyone's intuition all that much more. Regardless, the hope I have is that even though something initially seems really counterintuitive, that we can have such confidence in a result for a vaccine when there are so few people in a trial that get infected, well, you now have a better intuitive grasp of why we can be confident in those particular results. Finally, I love playing with simulations to help me get more intuition than I have just typically staring at formulas. If there's a version of this simulation that you'd like to see, maybe uneven sample sizes across vaccine and placebo conditions, larger or smaller sample sizes overall, leave a comment below, and I'll see if I can run that for you, and we'll post what I get in the comments. As always, thanks so much for watching.